Deconvoluting inflammation and immunology for clinical trials. That's our topic today, sponsored by Agilex Biolabs. And I'm delighted to introduce Kurt Sales, Director Immunoassay at Agilex Biolabs. Kurt's a pro at this, and he's got an info-packed presentation to give today. We're going to follow up with a Q&A at the end. If you have any questions, please submit those using the Q&A button below. I'm looking forward to seeing all of your questions on. Okay, let's start over again. See, I did mess it up. I'm going to start from the top. Deconvoluting inflammation and immunology for clinical trials. That's our topic today, sponsored by Agilex Biolabs. And I'm delighted to introduce Kurt Sales, Director Immunoassay at Agilex Biolabs. Kurt's a pro at this, and he's got an info-packed presentation to give. We'll follow up with a Q&A at the end. If you have any questions, please submit those to us using the Q&A button below. I'm looking forward to seeing all of your questions on today's topic. And now, without further ado, here is Kurt Sales.
Thanks very much, Arsalan, and hello viewers, and welcome back to this webinar series that we've put together. Hopefully today will give you a little bit of information around inflammation and immunology for clinical trials and a couple of different platform choices that are available for us. So just by means of contents, I'm just going to run through the Australian landscape, a snapshot of why people bring their clinical trials to Australia, an overview of our laboratories and our services, and then we'll launch into the topic for today on deconvoluting some inflammation and immunology for clinical trials. I'm only really going to focus on two aspects, a flow cytometry panel, it's just a general TB and K panel, and it'll be a little bit clearer as we work through the slides why I chose this as an example. And then a custom inflammatory panel using the Luminex platform where we investigate the responses of a custom panel in COVID matrix versus normal healthy matrix. So why do most people choose Australia for their clinical trials? Well, in short, the very fast startup is very lucrative for clients bringing their trials to Australia. So for example, a full CMC package isn't required. An investigator brochure, which details all of the preclinical data, supportive data, and insurance certificate and a protocol are all that's really required in order for a clinical trial ready to get started up. And everything lies within the ethics committee to evaluate the entirety of the data, to evaluate whether there's any risk to first in humans. So that means that first in human studies can start in as little as 4 to 6 weeks after the ethics committee submission. The data is that's generated in clinical trials in Australia is of the highest quality, and it's acceptable at all the regulatory agencies globally, including the FDA, EMA, MFDS, TFDA, and RCH. Importantly, for those companies that are earning less than $20 million worth of revenue, is an attractive research and development rebate of around 43.5%, which can be applied to the entirety of the clinical trial. So those companies that are in the burning rather than earning phase of their product life cycle can actually get almost half the money back from the Australian government for all of the clinical trial activities that are conducted within Australia. Important around COVID, we've been business as usual with all of the phase 1 units and business centers open throughout the entirety of the COVID pandemic. And this has really enabled us to move first in human studies through the pipeline and a lot faster. In terms of the bioanalytical, we can assure fast startup activities for both small and large molecule studies, as well as fast turnaround time for sample analysis to enable that adaptive design to progress for a SAD/MAD study to enable almost a week about dosing and speed things along. So the ecosystem comprises of a variety of different moving parts. We are just one component as a subsidiary of the entire clinical trial. And so we take care of the bioanalytical component for a lot of the CROs that we work with. I've listed some of them on the slide. We work globally, and we work with all of the CROs within Asia Pacific and outside. And the ecosystem comprises all of these components that need to be carefully considered for startup of the clinical trial. So it is a huge undertaking, but the CROs are really expert in being able to manage the clinical trial and put a package together. Importantly, a lot of the CROs can do a feasibility assessment, especially for those studies that require a disease population that might be a little bit rare. So a little bit about Agilex Biolabs. We really are the leading bioanalytical laboratory in Australia with over 2 decades worth of experience. And we work with global biotech clients. We are a specialist service center within the Asia Pacific, but we do phase 1 and also now global phase 3 studies that are hubbed in from various central laboratories or repositories into our lab for analysis. Our service lines comprise of 2 major areas: a small molecule tandem mass spectrometry area that focuses on all novel chemical entity and small peptides, and then an immunoassay and immunobiology service area that focuses on pharmacokinetics, immunogenicity, biomarkers, and pharmacodynamic assessments. Biomarkers overarchingly through both service streams, depending on the nature and sensitivity required for the analysis. So we're an FDA inspected laboratory. We're OECD GLP compliant and NATA 17025 accredited. And we work the same quality system for all of our projects. And the GLP accreditation ensures that we can service not only preclinical animal studies, which we do from time to time, but also the clinical studies, which focuses around 90% of our business service area. So what do we do? So the main service areas are basically put on the slide. PK assays, from either transferring an assay from a CRO or setting them up from scratch at both small and large molecule arenas. Immunogenicity assessments for large molecules where we use a tiered approach, and we can provide expert discussion around the specific nature of the immunogenicity assay that's required, where either as a standard homogeneous bridging assay or where high drug tolerance and sensitivity is required for those immuno-oncology studies where you're dosing a really high dosage of drugs as you move through the SAD-MAD phase. We often then go with a non-standard approach, such as a precipitation and acid dissociation, affinity capture elution, or a solid phase extraction with acid dissociation, the speed assay. We can provide neutralizing antibody assay support in both the ligand binding and also cell-based options. And more importantly, for those customers that are wanting to delve a little bit more deeply into the mode of action of their drug, we can support pharmacodynamic assays by either flow cytometry or cell-based assays. So we can provide a variety of immunological support, and I'll take you through some of the examples of this in the next coming slides. Where we don't currently have service areas, we partner with global laboratories in order to provide a full service option for our customers. So we really pride ourselves in being an end-to-end solution for regulated bioanalysis in the Asia Pacific region, but we're not only constrained to the APAC region, but we also work globally as well. So what do we measure and how do we measure it? So if we consider a clinical trial, and in the top center, the clinical trial starts with the subject or the patient. The drug is dosed into the patient, and then throughout the course of the clinical trial, blood is sampled from that patient or subject. Then the blood is then sent to clinical laboratories for safety assessments. These are standard clinical chemistry and hematology panels, but the blood is also sent to specialist laboratories such as ours, where we are doing highly specific and regulated bioanalytical measurements. And so the whole blood can be separated to serum or plasma, and we can measure the analyte or the drug in serum or plasma, either by tandem mass spectrometry or ligand binding assay. We can isolate PBMCs from the blood, and we can then expose these to an ex vivo culture, stimulate them with a mimetic such as PMA, ionomycin, and we can then create an ex vivo cytokine release assay in order to determine whether the drug has any immunological impact or alterative function on the immune system. And we can measure that using a variety of common immunoassay platforms, and I'll go through those options on the next slide. Importantly, we can also tag these cells with fluorescently labeled antibodies, and we can measure changes in cell surface markers in treated versus untreated cells, for example, and we do this using our state-of-the-art 5-laser BD Symphony A3 20-color flow cytometer. And this really is a powerful tool for multi-analyte analysis on single cells. And then finally, And cell lines in repositories that grown in cereal culture came from humans and animals originally, and so cells can be harvested from tissues, and they can be grown in cereal culture in order to create assays such as neutralizing antibody assays, ADCC assays, and then importantly, these cells can then be labeled with fluorescent antibodies, and you can measure endpoints by flow cytometry, or you can measure biomarkers using a ligand binding platform. So what are the most common platforms used in immunology and inflammation research? The most powerful for a broad spectrum whole blood cell investigation really is flow cytometry. It's a very powerful tool for immunophenotyping, target engagement and receptor occupancy assays, proliferation and activation of cells, and intracellular signaling. And the pro, if you like, for flow cytometry is its powerful multi-parameter analysis that can be done on a single cell. So you can see single cell changes in shape, in activation and proliferation rates. You can investigate rare populations, and you can also do some biomarker assessment. So it really is a powerful multi-parametric analysis. And one of the drawbacks is it really relies on a single cell suspension, as the single cells really need to make their way through the path of laser light in order for us to create these measurements. And so it makes tissues rather challenging, not impossible, but challenging. So the softer tissues, bone marrow, thymus, spleen, these you can harvest cells from these more readily, but solid tissues, it often relies on you doing a digestion, which sometimes destroys the cells. In terms of blood, it really does rely on fresh blood and or PBMCs that have been harvested and frozen down. And I'll touch a little bit more on that in our worked example in the next couple of slides. We have a huge amount of choice when it comes to ligand binding assay types. And there's a lot of really high-end, sophisticated and sensitive equipment on the market now. So we really are spoiled for choice in bioanalysis. So I've listed the main 4 platforms that we use for biomarker assessment and inflammatory biomarker assessment in particular. So the MSD imager, the mesoscale platform really is an electric chemiluminescent platform. It's very, very well characterized. There's an extensive array of kits and assay options across multiple different species. They've just brought out or bringing up more enhanced options with the S-Plex, which is a really sensitive assay platform, and the V-Plex panels tend to be really, really well characterized. So this is a company that spent a lot of time actually on characterizing their reagents and the kits, and we actually use this platform quite a lot, certainly for our immunogenicity assessments, but more often than not for our biomarkers. Similar to MSD is the Luminex. We've got a MagPix, and this allows multi-parameter biomarker analysis of up to 50 different analytes. There's a lot of extensive selection of kits available from multiple vendors. So unlike MSD and Quanterix, where you really need to buy a specialized equipment from these vendors, Luminex, there are a lot more choice available in terms of reagents. The drawback is it really does require a well-characterized and validated antibody pair from the vendor that you choose to buy your biomarker assay from. Probably last on the list, I put ELISA. Now, this is one of the oldest known platforms available. And we still use a single ELISA assays quite regularly for customers, and we validate these as well. The pros of an ELISA assay is it's quantitative. You've got a lot of choice of reagents. You can set these assays up from scratch easily in your own laboratory and validate them according to the EMA and FDA bioanalytical guidance documents. The drawback is you can only really measure one analyte at a time. And also the analytical range, because it's colorimetric, tends to be a little bit more truncated than the more sensitive platforms such as Luminex and MSD. In terms of sensitivity, I mentioned the MSD S-Plex, but the Quanterix platform really wins out in terms of sensitivity, and there's real focus on the lower end of the curve. And so where you might be conducting a phase 1 study or a healthy volunteer study, and your drug essentially reduces the inflammation, reduces the biomarker in serum or plasma. You really want to have the most sensitive platform that you can possibly find. And there are constraints because not every laboratory has all of these platforms in, but it really is key to be able to understand the data at the end of the day. So it's really important to establish the context of use of these biomarker assays, the indicative analytical range. And as I said, this is of particular importance where the study drug reduces the level of the biomarker in the human. So I thought I'd launch into our topic for today, so inflammation and immunology, and just give you a broad overview of what this essentially is, and then move into our worked example. So in higher organisms, the immune system is responsible for surveillance and detection and eradication of all foreign cells and invading pathogens. And it's comprised of a complex network of cells which start out as bone marrow-derived hematopoietic or progenitor stem cells. And these differentiate into the different cell types as I've listed on the slide. So lymphocytes, which are T cells, B cells, and NK cells, in monocytes in the bone marrow, and also in the lymphoid tissues and organs. And these cells are commonly called white blood cells, and that they are detectable in whole blood, and they can be isolated from whole blood and stored long-term as peripheral blood mononuclear cells, or PBMCs. These can be stored in liquid nitrogen for years, and they can then be resurrected, and you can conduct a variety of experiments using these. And for all intents and purposes, they still contain the characteristics of the individual from which they were derived originally. And inflammation really refers to the biological response to insult injury and involves these resident and recruited immune cells which are mobilized to the site of inflammation and injury and repair in response to chemoattractant stimuli. So these immune cells will churn out cytokines and chemokines, and they will then cause various immune cells to be recruited, extravasate through the vasculature, and mobilize into tissues where they would then mediate their repair or eradication of foreign pathogens. So I've got two case studies, one flow cytometry and then another one Luminex. And the first one, we investigated a BD Biosciences TBNK panel with absolute counting beads. Now this is a pretty standard panel. It's a 6-color multi-test kit, which is IVD and CE designated, and it's clinically used in a lot of the diagnostic laboratories for monitoring CD4 positive, CD8 positive, CD19, CD56 NK cells, and specifically for immunology and virology. This is a standard panel that's used to determine whether these parameters change in individuals during a clinical trial. These kits are really qualified and validated for specific bioanalytical platforms. So the FACSCantor and the Lyric in particular, this kit was validated for, but not our BD FACS Symphony A3 flow cytometer. Now, you would think all flow cytometers are the same, but they're actually not. Now, laser configuration is slightly different from the Canto. So TBNK cells, as mentioned in the previous slide, are the 3 major populations of immune cells in circulating blood. And CD4 T cells are helper T cells. They modulate the immune response by cytokine secretion, and they've got an indirect role in cell death. And CD8 T cells, they contain granzymes and porphyrin, they directly kill cells. And then, of course, B cells are responsible for those humoral antibody-mediated immunity. NK cells, non-discriminate, they're natural killer cells. They go around and they kill. So they respond pretty quickly to a foreign antigen that's invading the immune system. So we set this panel up to evaluate its performance on our FACS Symphony. And we used tree count beads in order to look at absolute cell counts, both before and after staining. And basically just determine the intra-assay precision of this kit and sample stability. And I'll go through that in more detail. So what did we do? So we took 3 healthy subjects. They were the same healthy subjects that we bled serially over 3 different days. And so whole blood was collected from these same subjects into K2 EDTA tubes over the 3 days, or on day 1, they were also collected into transfix tubes. These are cytomark tubes. Now, these are tubes with a fixative in them, and they are validated for certain cell types to be stable for up to, or stabilize them for up to 2 weeks. And we also used TrueCount beads from BD Biosciences. So we brought these tubes in, and they were used to provide absolute cell count analysis. So essentially, the experimental procedure is really, really easy using this kit. Took 50 microliters of whole blood into each TrueCount tube, added 20 microliters of antibody mix. This kit comes pre-mixed with the antibodies, 450 microliters of Faxlyze, and then whacked it on the flow cytometer and got our readout. So pretty simple. As I mentioned, we used the FACSymphony A3 5 laser 20 color instrument, which we had custom built for our specific purposes. And we set a stop gate at 2000 beads. Now, what I'd say is that the package insert says the compensation is not required, and this might be, appropriate for the lyric and the canto. But I'll show you an example where we actually had to apply compensation in order to linearize the data in our case. So we used analysis templates to set the gates for both experiments, and then we looked at percent parent with the percentage CD4 45 positive lymphocytes for the cell populations. And at the bottom of the slide, basically, is just the panel with the associated fluorophore clones. So the gating strategy was set in a hierarchical manner, gating off all events in CD45 lymphocytes, and then moving down the lineages from there. And we gated the counting beads separately, as shown in the bottom slide. So we got really good differentiation of all of these cell types, and got really good tight data, as I'll show you in a second. So I mentioned about the compensation. So, compensation is really essential when fluorophores are used, and especially those that use the same laser, because you get spillover into other channels, and that tends to skew the data. And as I said, the TBNK package insert said that compensation was not required, but this kit was really validated by BD Biosciences for the FACScanto and the Lyric machines. And we really needed to improve the discrimination of our populations. And so we applied compensation settings. We usually do this as a standard, but this just proves on the data plots on the slide that the notable shift of populations, in CD8 towards the CD4 gate. And this really may lead to difficulties in consistently identifying these cell populations. So it's important when setting these assays up to not believe everything you read on the package insert, and actually do the due diligence and do all of the tests in your laboratory in order to get the best possible data. So one of the drawbacks of the pre-mixed kit is that you can't do FMOs because everything is already pre-mixed in the packet. But what you can do is you can optimize the dilutional series. So we took our antibody mix and we then diluted it out from neat to 1 in 2, 1 in 4, 1 in 8, and so on. And we found that the optimal dilution really was 1 in 4. We got really good discrimination of the cell populations at that point. And after 1 in 4, the discrimination became a little bit more difficult to discern. But 1 in 4 meant that we could stretch the kit a little bit further and still get really good data. And this shows graphically all of the plots and compensation versus uncompensated on the right-hand side. So you can really see the CD45 spillover into the CD4 channel over here. And so it's really important to apply the compensation settings to the panel in order to linearize this and drop this back down again and remove the spillover into this channel. So we used the Cytofix tubes, and interestingly, cells shrink when they get fixed. So the side scatter and forward scatter plots needed to be regated on the fixed versus unfixed samples, and this is just another example of optimizing the conditions of the experiment right at the beginning in order to really be able to differentiate out those cell lineages. So we looked at intra and inter-operator precision. As I mentioned, we had the same three subjects that we investigated their blood over 3 days, and used this study. We used a single operator, K2 EDTA tubes, and we basically took a new. Each subject was sampled every day, and so what we have is a percent CV, either percent parent or absolute cell counts, from those 3 individuals per day. And you can actually see that even though we've got really good less than 20% precision within the K2 EDTA tubes, the transfix tubes, where we actually took the sample once, so we had 1 tube and we just aliquoted 50 microliters from that same tube each day and redid the analysis, we got really good consistent data. So the individual day-to-day variances in immune composition actually drives quite a lot of change within these cell populations. And that's to be expected as immune cells undergo a diurnal but also environmental change within different individuals. And then the data on the right showing the same experiment, but with the true count beads. And again, transfix tubes providing a more sort of consistent result day in and day out. Inter-operator, very similar, where we analyzed the 3 samples on the same day of 2 different operators, and got really good tight CVs in both percent parent for the transfix and K2 EDTA tubes, and slightly more variable with the absolute counts of the TrueFix tubes. So on the whole, this panel behaves really well and really consistently, and we're quite happy with it. We then had a look at post-stain stability. So what's really important when setting up these experiments is to try and cater for all eventualities. The power might go off, even though we have uninterrupted power supply and a diesel generator as backups in our labs. Sometimes a cytometer can blow a laser, and it might be several hours before you can get an engineer out to come and service or replace it. So it's important to have a look at some post-stain stability on samples to understand exactly how long you can stretch the analysis out before the sample becomes pretty much useless. And so we took our tubes and we fixed them. So we used Cytofix from BD Biosciences, and we did the analysis at time 0, and then put the tubes in the fridge, and then redid the analysis after 6 hours and then the following day. And so interestingly enough, we got really consistent data at 6 hours. The CD16, CD56 channels started blowing out a little bit, and these are more volatile cell populations. But really, after 24 hours, the red bar chart shows that your experiment is pretty much kaput at this stage. So 6 hours post-stain stability we found acceptable, was within our 20% criteria that we had set for our experiments. And then 24 hours, it was much greater than that for all cell populations. So that was sort of fixed sample stability. But what happens to samples over a long period of time? So we're working in a clinical trial setting where, even though we're situated in Adelaide, which is readily accessible from all of the phase 1 units throughout Australia and New Zealand, and we're working with a phase 1 unit in the Philippines as well, we can get blood samples into our lab within 24 hours. But what's really important is to actually understand what absolute stability you will have over a period of days. And so, so what we have in this experiment is we have the N of 3 tubes where we have analyzed the same tube over 7 to 10 days by a single operator. The K2 EDTA tubes, basically they were stored on a rocker at room temperature throughout this period of time, and the transfix tubes were basically stored at 4 degrees. And so what we're trying to do is to mimic what would normally happen to a sample that is in transit from a phase 1 unit that might get stuck somewhere. So it might get stuck in customs at room temperature or ambient shipping temperature. What happens to those cell populations? And so on this slide, you can see that the K2 EDTA and transfix tubes, the stability was really good for the CD3, CD4, CD8 populations for pretty much up to 10 days. So so these are more robust cell types, and so the indicative stability is greater. The transfix tubes, which I said adds that fixative, and it stabilizes those cell populations, and for these populations, the tubes are validated for up to 2 weeks. So unsurprisingly, we got really good data for those populations. But on the lower 2 charts, you can see the fallout with the CD16 and CD56 cell marker population. So the NK cells that were a bit more of a volatile cell population, they, after day 1, started dying off and were of no good. So the take-home message of this slide is for rare populations or populations that are a bit more volatile, a fixative tube, transfix tube such as this might be really appropriate as the blood collection tube. Now, these fixative tubes have a drawback because you can't do cell stimulation experiments because essentially you've added a fixative to it. But for broad spectrum immunophenotyping, you essentially are fixing the blood population as it comes out of the subject, and I believe will give you a more robust data over a longer period of time, and also take care of that contingency for something happening along the way with the sample. We found very similar with the absolute cell counts, so using the true count tubes, and this the data basically mirrors what we found for the percent parent. Not quite as tight data, but certainly the transfix tubes really win out again for both the true count, absolute cell counts, and percent parent for this particular panel. Over 10 days, and in particular that CD16-56 cell marker population. So the take-home message is that the panel behaved really well, but we needed to do a couple of tweaks compared with the package insert, such as apply the compensation settings, and then we also, for our own requirements, found that we could only really apply post-stain fixation for up to 6 hours, and then collecting the samples into transfix tubes improved the stability for up to 10 days. So really is a powerful multi-test kit for a simple workflow and analysis of those major immune cell populations, and something that we'll start to be using a little bit more of as a general panel. So moving quickly ahead to our multiplex COVID-19 panel. So we evaluated this custom Luminex panel from R&D Systems. So SARS-CoV-2 virus caused a major disruption globally over the last 12 months as a result of the global pandemic. And what it does is similar to many other respiratory viruses. It infects the lung, causes massive immune response and tissue damage, and in some cases even death. And so we were quite interested in the flavor of immune cells and inflammatory markers that might be heightened in subjects with COVID-19. So we are doing quite a couple of COVID trials at the moment for a variety of different novel drugs and indications. And so we looked at the literature to try and get the best bet on what biomarkers might be most abundantly expressed. And so, it was the usual subjects that came up. Quanterix also did a really good webinar, and they've got some fantastic high sensitivity panels that are available now as well. And so what we did is looked at all of the data, and we came up with this custom 10-plex panel from R&D Systems. And this really comprises interleukin-8, GM-CSF, alpha and gamma interferon, IL-12, B70, IL-17, IL-1 beta, IL-3, IL-6, and TNF-alpha. So a really broad spectrum immune cell panel, which we thought would be applicable not only to COVID, but perhaps also to other inflammatory disorders that we are currently working with sponsors on a regular basis. So we just used a standard workflow for this approach. We tested 86 samples in total. 41 were healthy volunteers, so this was normal healthy serum. We bought our serum in from a serum repository or serum supplier, and we had 45 COVID-19 positives. 35 of them were serum and 10 were plasma. So we tend to do our biomarker analysis on serum more often than not, as we just find it provides a clean response, as you don't have the matrix interferences with fibrinogen, etc., that you do have with plasma. So this table just gives you the data on what we got, healthy population versus COVID. So as to be expected, healthy population, you're going to have very low levels of inflammatory mediators unless there's something wrong. And so the only biomarkers that actually gave us a positive signal above BLQ was IL-8, IL-1 beta, and IL-6. Whereas with the COVID positives, we had quite a considerable response for all of the 45 that we investigated. We then had a look at the difference between serum and plasma, and this was quite a stark difference. Is that for the and yes, so there were only 10 plasma samples, but for the 10 plasma samples, some of the biomarkers were actually BLQ in the serum versus plasma, and these are taken from the same person. So so it just gives you an indication of the biological matrix can actually be quite important when it comes to clinical trial design and determination of those biomarkers, and also the platform of choice. So just looking at the distribution of those three that we could pick out in the healthy population versus the COVID, and you can see that the differential distribution in the COVID in particular. And so we had a look through the information that we got from our repository on the patients that they had selected these specific matrices from. And there was an indication, and the N number is probably too small to actually get a really confident about the supposition, but that those biomarkers that were actually highly expressed in these three in particular tended to be those COVID patients where the serum or plasma had been harvested early on, or early onset of the COVID-19 inflammatory response. Whereas those subjects that were COVID positive and that might have been 2 weeks rather than 8 days past diagnosis tended to be at the lower end of the curve. So there would be an indication that that early onset of COVID, you get this heightened inflammatory response, and then it drops down a little bit after that. So I suppose the take-home message here is that when ordering matrix from a supplier is to try and get as far as possible a similar time of onset to reduce variability, or if we're wanting just to have a normal distribution and an average across that, then by all means go ahead and get a selection such as what we did. So really one of the take-home messages for us was that we really needed a more sensitive assay, especially for those healthy volunteer subjects. So, a lot of the time we'll be looking at study design where there'll be a placebo versus dosed, or healthy versus disease, or it might just be a healthy volunteer study where the drug is reduced the concentration of biomarkers. So when you're starting out from a level of detection where you can't actually see anything on using an assay kit that already has a relatively good lower end of the curve sensitivity, you're not going to see anything when the study drug reduces those biomarkers even further. So looking at the various suppliers, so going right back to my first slide, one of the first slides is looking at options from Luminex, MSD, Bplex, and Splex, and then the Quanterix. You could see that the panel that we chose was probably the least sensitive of all of the platforms. Had we gone with the MSD, we probably would have had a few more data points on the curve for those that were BLQ. Certainly, if we'd gone with the Splex, we would have actually most likely had most of them on the curve. And then the Quanterix Simoa platform, is, as I mentioned before, really highly sensitive. So, so when it comes down to, the study design, these are really important considerations to discuss with your bioanalytical provider. We also need more well-characterized samples in order to identify exactly the levels of COVID, and to actually try and come back to that context of use of, in a clinical trial setting, what is the N number that we actually need to put through the qualification of the assay in order for us to set the acceptance criteria for an absolute change that is required in a clinical trial setting. So just to conclude, few biomarkers are detected in healthy matrix, and we detected the majority of those biomarkers in COVID matrix. There was a difference in serum versus plasma, which we found interesting. But as I just mentioned, the platform and assay of choice is really important when designing the study, as the lower limits of quantification are really important considerations for for getting really good, robust, and reliable data. Happy to answer any questions, and I hope that you found the webinar somewhat interesting, and we're happy to be contactable in order to assist you with your next project. Thank you very much.
All right, thank you, Kurt. Deep expertise as always, and I know the audience appreciates that. So here's some questions for you. Let's start, here's a basic question. What are the main considerations for biomarker assays to support clinical trials, Kurt?
So really the main consideration is the context of use of that biomarker assay. So what kind of data is the sponsor wanting to get out? Is it just a case of, we're worried that we might have an inflammatory, an infusion-related reaction or something, and we just want to broadly monitor whether the inflammatory markers would go up? So we're not really worried about it being BLQ. We're just worried about if we're going to detect it. Then that's one consideration. If the biomarker is actually being used as a pharmacodynamic assessment, so the biomarker is affected by the drug, or it's a very important biomarker in that clinical patient setting, and going to be used for decision making, then it really is critical to set up the most sensitive and appropriate assay so that the linear range is appropriate for the entirety of the trial. So if it's a SAD/MAD, you want to be able to be within the linear range of the curve all the time without having to try and guess when, what you're going to have to dilute the samples in when they come in from the trial. And then also it's to understand that the day-to-day or patient variability that you would likely get in with that biomarker. So biomarkers undergo diurnal and nocturnal changes, and there's of course individual differences within a normal healthy and also diseased population. So it's really important to really understand what you're actually looking for in terms of data to get an idea of what the variability might be when we set that assay up, in order for us to be able to deliver data that is really going to be robust and stand up to regulatory scrutiny.
All right. How about this then? What are the main considerations for sample integrity for flow cytometry assays?
Yeah, so that's a very, very good question. So it really depends on what the cell populations are that are of interest, where the clinical trial is being conducted, and the likely route of transport from that clinical trial phase 1 unit or patient clinic and into the bioanalytical laboratory. We do everything fresh as it comes in to us, but there can sometimes be a delay. And what we need to consider right up front when setting the method up is to add on those stability assessments so that we can get a really good indication of what happens to the cell populations over time. And then whether there is any benefit in using a stabilization tube, such as the Transfix tube, for the clinical trial setting. So Transfix tubes are fantastic for those broad immunophenotyping panels, a panel that might have Ki-67 or proliferation added to it, because really is fixing the cells as it comes out of the patient setting. It's not the holy grail. There are drawbacks to using this type of tube, and it really needs to be well considered within the context of the assay setup. But certainly, being able to identify the changes in the cell populations over a specific period of time will then really guide, your experimental design, and whether. So we can still see changes in a clinical trial setting, after several days if we're looking at percent M5 and percent parent. And we gate off the dead cells, and so we're only looking at the live cells going through the channel, and we can use several dump gates, and so we can actually utilize blood that is a little bit older and still get some meaningful immunological data when it comes to receptor occupancy or such like. But really, when it comes down to those broad immunophenotyping changes, then a little bit more consideration needs to be had on method development and then setting the panels up appropriately.
Okay. All right. And I got one last question for you, Kurt, for today's session. So you mentioned several platforms for measuring biomarkers, from Luminex to MSD to Quanterix and ELISA. So which one of these is the best for biomarker analysis?
So again, it comes down to the context of use, the type of biomarker, what is available in the literature around the levels that we could expect. But generally, it comes down to the sensitivity that's required for the biomarker. So, as I've mentioned, if it's a healthy volunteer setting, and the biomarker is not going to be detectable during using the classic forms such as an ELISA or maybe even an MSD Bplex or Luminex, then the only option is Quanterix, but not all laboratories have all the platforms available. So it then really becomes a discussion with the sponsor on, do you really need this assay as part of your clinical trial when the data is at the lower end of the curve, where it's not linear anymore, and small changes will manifest big CVs, and you're not really going to get really good data out. That becomes an important consideration. So it's really looking at what sensitive options are available, but then looking at more closely looking at what the absolute requirement is in the clinical trial, and whether it's a disease population and what the drug particular is going to do to that biomarker, to get some to make them get the most information out. So these are often questions that we will ask when we get a request for proposal. Is basically what is the study, what is the drug doing to the biomarker, and what changes does the sponsor think they want to see based on their preclinical or in vitro data, monitoring those biomarkers over time. So, more often we'll see biomarker assays just added in as optional extras without much thought about what the data is really going to be used for at the end of the study. So it becomes really important to understand exactly, the ethical considerations of taking that blood sample from the patient. What are you going to actually use the data for in terms of those biomarker assays?
Okay, all right, Kurt. Well, that's all the time that we have for today. Kurt Sales, thanks as always for sharing your knowledge and your experience with us. Always enjoy doing these with you. And thank you to Agilex Biolabs for sponsoring the topic. If you want to
Thanks.
rewatch this webinar or share it with colleagues, a link for on-demand viewing will be sent tomorrow to anyone in the audience. So I'm Arsalan Arif for Endpoints News. Thanks for joining us, and we hope to see you at a future webinar. Thanks, everyone.
Thank you. Thanks, Arsalan. Thanks, viewers.